We're going to be doing some protein expression, and it's really important that when we do so, we add the appropriate antibiotic to our media. And so we use antibiotics as selection markers to select for, or basically only allow to grow, bacteria that have actually taken in the plasmid that has the instructions for making our protein. So basically we recombine the instructions for making our protein with a plasmid vector backbone, and so we got this circular piece of DNA that we can put into bacteria, and it contains the instructions for making our protein, and the bacteria take it in and then they make protein from it. Now, some of the bacteria aren't going to take it in, and some of the bacteria are going to take it in and they're going to lose it because these plasmids are hosted extrachromosomally. So basically the bacteria doesn't incorporate it into their own DNA, they just keep it outside of itself, and so then if the bacteria divides, they might lose that plasmid. And if they lose that plasmid, well, now they've got less of a burden, and so they might even grow better. So we need to give the bacteria an advantage if they have the plasmid. And the way that we do this is by giving the other guys a disadvantage. So in our plasmid, in addition to the gene for making our protein, or more technically the CDS, the coding sequence for making our protein, we also have an antibiotic resistance gene. This way, if the bacteria have the plasmid, they're going to not just make our protein, but they're also going to make a protein that gives them resistance to a specific antibiotic. If we add that specific antibiotic to the media, so the cell food that we're growing these bacteria in, then only the cells that have the plasmid that has the gene for your protein will be able to grow. And all those other bacteria that either come in from the outside, they never took in the plasmid, or they lose the plasmid over the course of their growth, are not going to be able to grow. And so we select for the cells that actually have the plasmid. And this is great because now we're only feeding the cells that are actually going to hopefully be able to be making our protein. It's important that we can then add the antibiotic in whatever culture media we're growing it in once it's taken in that plasmid, so that we keep alive the ones that keep the plasmid and kill off any that lose it. It's also important too, though, that you don't add your antibiotic before you autoclave things, because then the antibiotic is just going to go poof. And the antibiotic is going to kind of go bad over time in the media, and so you want to add it fresh to the media right before you add your bacterial sample, and then you don't want it to like overgrow and stuff, or else the antibiotic goes bad and the bacteria can go bad.